Hello and welcome to my interview with the popular YouTube poster Concordance. Thank you for joining me.
Happy to be here.
Now, Concordance, you do videos on many different topics, but your actual career is in virology, correct?
It started out in virology. I've since branched out to a lot of different areas. I don't work in academia anymore. I left academia several years ago. So now I work for corporate paymaster and I do what they tell me to do. And that includes a lot of applications development. Virology is just my one true love.
I see. And now, in a recent video, you said that you were going to the American Association of Cancer Research meetup. How was that?
It was excellent. For the third year in a row, the hot topic was cancer stem cells, and that's a topic I'm kind of interested in. We had a chance to present a poster there. I kind of got roped into having to stand next to a poster, which I haven't done in almost 10 years, while other scientists walked by and asked me questions about someone else's work, which I wasn't too happy about. But got a chance to visit Orlando. The weather was very nice. I got a chance to meet a few really interesting people. So overall, it was a pretty good meeting.
Well, it sounds like a fun time. Now, it wasn't necessarily your work, but was this your first time presenting?
Oh no, no, no! I've given very minor sessions. I've been on a review panel before, but I haven't been in cancer research very much in the last few years, and I haven't had to stand next to a poster for a long time since I was a grad student.
I see. Now, you're probably tired of explaining it, but could you give us a little bit about what kind of research you were presenting?
In this particular case, it was a methodology paper or a methodology poster. So we were talking about a fairly new way to measure the modification of genes. So the genes themselves, of course, have C in varying amounts in particular sequence, but then the little DNA molecule also has some modifications to it, so it can be methylated. And if it's methylated, it's either switched on or switched off, depending on particular context of the cell. So we're looking to see whether those switches are on or switched off using a PCR-based methodology. So it's a new type of assay for methylation. This relates back to cancer research in that in a lot of cancers, there's abnormal methylation state. And a lot of cancer drugs are focused on changing that methylation state, either usually demethylating, so turning the switches off, quieting the cancer cell. So there's a lot of interest in whether or not that's working and whether or not it's affecting the right genes or too many genes, genes we don't want to affect. So being able to measure that accurately is key to developing better drugs.
I see. I'll pretend that I understood most of that. And now you say cancer research hasn't been your main focus for a little bit. What has been your main focus, if you don't mind me asking?
Like I say, I do what they tell me to do. I develop applications for technologies that my company owns or makes. And so I've worked on a pretty broad range of different problems. I'm one of those rare scientists that is not hyper specialized. So most recently, we've been working on probiotics. It's a very hot topic in a lot of areas. So we're looking at being able to measure what's called the microbiome, which is the diverse little communities of bacteria that live inside your body. The reason why this is really important is you look at some people, for example, who are obese. They have a distinctive microbiome, a particular mixture of the little bacteria that live in their guts. And it could be that we could predict or treat obesity or diabetes or cancer or a variety of different autoimmune problems by making adjustments to that microbiome, or at least understanding what it is about those bacteria that's causing the problem.
Now that I understood mostly. Before we switch gears, the corporation you're working for won't sue me, will they?
Possibly. I think they have more lawyers than scientists these days.
So just like there's the Discovery Institute.
Oh, yeah. Yes, they absolutely will sue you if you say something negative or if you reveal anything proprietary. They'll
Okay.
also fire me, which is part of the anonymity of YouTube.
Okay. Well, let's switch gears to YouTube. As I said, you've covered many different topics, but what do you see in the future for the Concordance channel?
There's a lot of interest right now in drugs. God, how I wish I hadn't made that cannabis video. Wish I hadn't ever mentioned anything about cannabis because it's probably 30% of the emails or personal messages I'm still getting are specifically about what an idiot I am and how I don't know anything about research or cannabis and how cannabis isn't addictive and how it doesn't have any negative health effects. So there's been a lot of push for more information about illicit drugs or harmful drugs. And I'll probably focus on that a bit. I also would like to talk a bit more about my research. There's a project ongoing with NASA that involves putting some equipment up on the International Space Station. And they want the equipment essentially custom designed, and I'm participating in the project to design that piece of equipment that will go up. And that's been very exciting. That's been going on for over 4 years now. There's nothing quite as cool that I've ever done as sending something up into space. And I've asked if I can sign my name on the side. They didn't like that idea very much. So I'll probably talk about that. I've got an essay queued up about the nature of physics and philosophy, and I've got a video queued up about MSG and aspartame. So those are always very interesting topics. What I found was there are enough atheists and there are enough non-theists on YouTube that talk about how science makes their life fulfilling. So I thought I'd try to fill a slightly different niche in talking about some other topics that require a little bit of debunking or a little bit of critical analysis. So I need to avoid using the word debunking because that's not really what my goal is. My goal is to shine a little light on them.
Well, it sounds like a very exciting stuff, and you certainly don't like to specialize, do you?
No, I don't. The easiest videos to make, and the most fun for me, are the read essays, the science essays from famous scientists in the past. It's kind of interesting for me to read someone else's words aloud and try to bring them to life, and then create the video portion based on photographs and historical records of their life and what they did. I've learned quite a bit by reading other people's essays.
Yeah. Okay. Well, I look forward to your new videos, and thank you very much for joining me.
No problem. Great to talk to you.